this is always a wonderful event. It never has the kind of crowd that we think it deserves, that we know it deserves. But we've gotten to be okay with that because the people that are here are good to be here, and the science that this represents is just kind of spectacular. As Monica sort of hints, I've been thinking for a lot of reasons of the early days of the epidemic. They were really exciting, really awful, but the progress that we've made has been just stupendous. The distance that we've traveled from those days to this one are indescribable. I think never before in any part of medicine have we seen such an amazing transformation. And I've got to get my phone going here. I got my notes. Screen went off. But you know, as much as I might kind of muck around in the early history of the epidemic, it is clear, I think, to everyone that there is a present and future for HIV research as well as a past. The challenge is not yet over. The research will look different for the challenges that remain, just as the researchers look different than people that have been around as long as I have. One of the, without any question, one of the real joys in having had a long career and having had the amazing opportunity to be involved with CFAR and to work with Warner has been getting to know really the next generations of leaders. And some of the people that have crossed our paths are now pretty senior. Some of them, I think, are now full professors of medicine. And to watch that happen, to watch the excitement of early careers, the brilliance of younger minds is really a joy. And with CFAR, we've had the chance to very directly support early careers, very intentionally. It's what CFARs are in large part organized to do. We give pilot grants. Our CFAR has made a special effort to give as much money as possible to pilot grants. Our success rate, your success rate, in turning pilot grants into K awards is really unmatched in the rest of the CFAR universe. No place devotes as much of the resources or has such a high success rate. Other CFARs don't allow postdocs and fellows to apply for pilot grants. They restrict it to people already on the faculty. We've kind of taken the opposite tact, and I think as a result have more success than anyone could imagine. So the work you're doing is really impressive, and in addition to the pilot grants, the mentoring program, as Jonathan and Monica have indicated, and as you know better than anyone, has been really at the centerpiece of what we've done. It's just such an amazing thing to watch the people come into the program, the mentorship that Jonathan and Monica provide, as well as the mentors that are matched. I think is along with the RAP grants really responsible for the success that you all have had. So that's been great, and we know it's a great program because every other CFAR is jealous of the mentoring program that we have. Jonathan and Monica are asked to come to other CFARs regularly to talk about the program here. In the Mentoring the Mentors program that they also do to train mid-career and senior investigators across the country has resulted in better mentorship in the field of HIV than would otherwise be the case. So I think our impact from San Francisco is really hard to measure, but it's a really very positive thing. So thanks to them. As Monica suggested, this is a period of transition. In fact, I think this week the committee will be interviewing candidates to replace, if that's possible, me and Warner. I think it is. We've threatened to turn over the leadership as our managers have learned for about the past 12 years, and this time we are finally doing it. We think it's really an important and great opportunity because, again, the kind of research is different than it used to be. I think it's appropriate to have leadership that is attentive to the future of AIDS research and the big steps now needed to really bring this epidemic to an end. I have promised not to be intrusive, but I hope to be around, and I won't be leading this symposium next year, but I'll certainly be in the audience and eager to keep track of the next generations of young investigators. So thanks to all of you for the work you're doing. I think the field remains really strong. It's, again, strong in a different way than it has been, but it's going to be great fun to see this progress. Let me mention Warner. I was the first, I'm the last remaining PI of a CFAR in the country. We were funded in 1988. Warner came in the early mid-90s and to lead the Gladstone at the General. And very quickly, we established a relationship and decided more or less organically that it made sense for us to be co-leaders of the CFAR. I remain the PI, but that's just for the paperwork. We decided early on that this really needed to be an absolute co-directorship. Warner and I meet very frequently. We meet with our managers. Now Lauren Sterling, who's been amazing, we meet as a trio very frequently. We don't make decisions by ourselves. We make it as a duo and trio, and it's resulted in our ability, I think, along with CAPS, to make sure that our attention on the entire spectrum of HIV research in San Francisco is really on point. Warner has been a great collaborator. He also is going to kind of not leave the scene. He's going to be the director of the Cure Center here at the Gladstone, and I look forward to continuing to work with Warner over the next however many years we're given. So Warner, welcome to the stage.
I decided not to go up on the stage. I was just going to sit here. More of a fireside chat. So well, first, Paul, I have to say, it was 92 that we joined forces and established co-directorship. Is this working? Yeah, I think so. Maybe I just have to, like American Idol. Yeah. Or The Voice. But 92, that's been a long time. And in fact, I was thinking about when did I really start in research? Well, it was in 1970. It was in 1979 I finished my training. So what I'd like to share with you is like 40 years of history, of experiences, boiled down into 10 take-home points. So the first thing I would say is make certain that whatever you do in science or medicine, clinical research, wherever you are, whatever you're doing, make sure and always put it continually to the test. Are you passionate about that? Are you really passionate about it? It's not enough just to like it. Are you passionate about what you're doing? And if you're not, find out what you are passionate about and do that. I cannot say that I think that I found myself occasionally veering away from that rule, always regretted it. The other thing that I think that I've learned along the way is work hard, play hard. You have got to achieve that kind of balance in your life. You can easily in this business burn out. I've seen it in multiple scientists. But if you're able to achieve the right mixture of kind of love of life and love of science, you're going to go a long ways. But you've got to make that, you've got to really make that a priority. And it's so easy to get sucked into the lab or into your work, but make sure you take time for yourself. I'd also say that consider the path less trodden. Too often in science I see we're almost like lemmings. We follow, we kind of follow each other, and we're all on the beach, and I just saw the baby iguanas getting eaten by the razor snakes. That's what it's like. I mean, think about a different. Well, there's the joke at Harvard. Your fellow professors stab you in the back. At UCSF, it's a much more collegial environment. They stab you in the chest. So, but back to the path less trodden. The path less trodden. So, yeah, don't always go where everybody else is going. In fact, there's tremendous biology. And you know where the richest biology is? It's at the intersection of different fields. So find those intersections. The people are not really working there. Find those. That's where the interesting science is often to be discovered. Now, you also, because you're going to be working at intersections or doing things that are different, you're on the different path, you're likely going to have to reinvent yourself 2, 3, 4 times during your career. You can't continually do the same thing. You've got to be prepared to reinvent yourself as a scientist, as an investigator. My 6th point, embrace new technology, but don't become a slave to it. So there's all kinds of new technologies that are constantly emerging. Be receptive. If that can help you do what you're passionate about, adopt it. But never allow the technology to be the major driver of your science. It's a facilitating role. Follow what you're passionate about. If it happens to be a new technology, that's fine. But do not allow the technology to dominate unless it's truly at your core. So number 7, be a good mentor. Frankly, your trainees are going to accomplish far more than my trainees will accomplish far more than I could ever accomplish on my own. And so your greatest legacy, your greatest impact in science is not what you do, but what your trainees do. And so make sure they're really well-trained and prepared for life in science. One size does not fit all, certainly in mentoring. I mean, I'm talking to the masters here. They have created a mentoring program that is, I think, unrivaled and highly desired, as Paul was mentioning, by other centers throughout the country. But one size doesn't fit all. You really have to take an individualized approach. And if I was a mentee, one of the first things I would do is find multiple mentors. I mean, I would seek opinions from multiple individuals and then synthesize the best advice from each into making your career forward. So be fearless about recruiting new mentors. That wasn't, when I was training, this whole idea of mentorship, it was more, it was like you were the Padawan, right? You were hoping to find the Force. People did not take an active role in mentoring. Sometimes you would find people who would be a little bit more generous, but in general, they were just, kind of role models to which you aspired. Now, though, there's a much more active exchange between senior and junior investigators. Take advantage of that. Seek out multiple senior investigators to inform your life. And then finally, I've often told this to my lab. Really to be successful in this field, almost at whatever you do, you've got to do 4 things well in academics. You've got to be able to present your work well. No pressure on the presenters today, but it is true that more times than not, people form a first impression of you hearing you speak. You know, and that is an indelible impression. Were you clear? Were you concise? Were you compelling? Did your slides work? Did you appreciate the fact that people zone out on average every 2 to 3 minutes? And a great talk allows people to climb back into the talk. Not something, have you ever been in a lecture where, and may I have my summary slide, and everybody in the room sits up because they had no idea what had happened for the prior 45 minutes. But here's my chance to really find out what was said. That's not a good talk. So constantly give a great talk. The other thing that you have to do is you have to be able to write. More and more writing is, it seems to be being de-emphasized, but it is critical. Being able to write a cogent scientific paper with a well-constructed introduction, results, discussion, realizing that only in the discussion do you have any latitude. So you have to be able to write a scientific paper that will stand the test of time. The other writing exercise that you have to do is write a grant, and that's completely different. You essentially, with a grant, you've got 15 minutes to sell your ideas to some reviewer, generally hostile in my experience, a reviewer who doesn't want to fund you. You've got to convince them that your idea is worth funding. So this is a marketing document. It's not a scientific paper. They're two completely different writing exercises. But in order to be successful in this world of science, you have to be able to do both. And then finally, and perhaps the most difficult skill that you have to acquire is knowing the difference between A and B science. 90% of the science that's published that you read is B science. And it's B science because that it will often not stand the test of time, that it's often poorly controlled, and that's why it doesn't stand the test of time. It looked good at the moment, but because the work was incomplete and not purely and completely controlled, there'll be another alternative explanation that will actually emerge as truth. So A science is so well controlled that there is no other interpretation, that everything has been thought of and the question is asked repeatedly in different ways and it emerges. And so be an A scientist. Now, how do you learn to do A science? In my lab, and I'm sure the people in the back who were in my lab kind of shudder at this thought, I found lab meetings, it was about 10 years ago that I found lab meetings that people were not asking the tough questions in lab meetings of their own colleagues because they didn't want to be too aggressive or critical of their lab mates. So I decided to try and depersonalize this. And so what I did was I could spend my life reviewing papers for journals. So I decided to bring a number of individuals in to review papers. So in general, we review papers as a group of 5 or 6 people. And we go over the figures very carefully. We talk about the key missing controls, why this experiment was good or bad, et cetera. And I have to tell you that in the end, it's like Shark Week. The postdocs and students in my lab have become so good at ferreting out the problems in a paper that sometimes I have to temper their enthusiasm for rejection. But it has been a useful tool to really train young scientists to be critical about work and be able to see what's missing and then to hopefully design their own experiments in a way where those key controls aren't missing. So that's it. My 10 thoughts, lessons learned over 40 years. It's kind of sad that that's it, right? But I do want to thank I want to again thank Paul and Lauren and all of our prior CFAR managers. It's been a wonderful time since 1992 as a part of our center. I think we've made a lot of progress, and really excited about the new leadership which will take the center to yet higher heights. Thank you.
you hadn't played so hard, though, you wouldn't have had to have all your joints replaced.
That's true. Basketball did me in, that's right. It's been a tough 6 months. I've had 3 joint replacements, 2 shoulders and an ankle in the last 6 months. And I've not been a happy camper.
In addition to your 2 knees earlier.
I am truly bionic. The best thing that's ever happened to me are these microwave scanners at the airport. I get right through them.
Thank you. Hi, thank you for the keynote speeches. I have a question for Paul. You mentioned that in the future, research and researcher will look different. Can you please expand on that?
Well, what I was really meaning is that they'll look younger. But on a somewhat more serious, although that's a pretty serious issue too when you get older, I think it's the attention that we're now giving to the recognition that HIV and a lot of the other problems that we face really can be seen just in scientific terms, but really should be seen in terms of the social setting in which they're occurring. I think the kind of research that's going to be really important for the next part of this epidemic is going to be driven by that. So thinking of the communities, thinking of the not just the big populations, MSM or whatever, but the more specific descriptions of people involved are going to be really important. And it's important that the researchers match the diversity of the challenge that they're dealing with. So I think it's encouraging. I haven't really sat down and mapped out. We don't do as well as we would like to do with diversity, especially African Americans. We just haven't had a great pipeline, but I think attention to that going forward is going to be really important. So I think it is important that they be young, but it's also, I think, important that they, whenever possible, match the complexity and diversity of the epidemic.
Maybe I'll just add a thought or two to the particular situation of basic scientists in HIV research right now. I think this is the best understood virus in the history of science, without a doubt. Within 10 years from the discovery of the virus, 10 years later, we had multiple drug antiretroviral therapy that changed the trajectory of the epidemic. It's a truly stunning progress. It'll go down in the history of medicine of science as a true milestone. But to some extent, now with over 30 drugs, we are, as basic scientists, we are the victims of our own success. We see, in fact, that NIH funding is no longer for really truly basic discovery work. It's more for applied research. It's for a vaccine. It's for a cure. Yes, there are basic components to that. But in general, the landscape is changing. And I think, talking about reinventing yourself, I think that most basic HIV scientists now are going to have to reinvent themselves. I've visited many universities who have not a single HIV investigator and no desire to recruit one. I mean, it's a changing landscape in terms of the basic biology. And to that end, it was about a year ago that I looked at the next 10 to 20 years of GIVI, the Gladstone Institute of Virology and Immunology, and decided that it was time for the Institute to pivot in a new direction, away from HIV. We're in the midst of that pivot right now. I stepped down as director to make way for a new director. I, of course, wanted to continue my career in HIV, and that's why I'm leading this Center for HIV Cure Research. But it is a changing environment for the basic sciences. It's much more now an issue of implementation science and very much translational applied research.
Other questions?
Thank you both for that keynote. And
I was wondering, and this may actually be sort of the corollary of what Warner already said, but as you look back at your very impressive careers thus far, are there things you would have done differently? Are there, with hindsight, are there lessons you wish you'd learned earlier that you can share with us that might help guide people going forward?
I'll tell you one thing. Relationships that you form along the way in your road in science, along that path less trodden, value, honor, celebrate those relationships. Never let them die. I think that you never know when those people will come back into your lives in a very meaningful way. So I think that I've not been as good at that as I needed to be. And that's something that I would change in terms of my career in science.
Yeah, so I guess I wouldn't say that I have major regrets. I loved what I've done, but I could have done it better. You know, and some of this is just, you know, when I was at the General, I think a little bit more attention to the finances of the operation would have been good. I think, so I think there are some lessons there that I'd be happy to tell people about. Yeah, so I mean, there's a lot of things that, you know, anyone wishes that they had done a little bit differently. People that I didn't keep track of, pay attention to. I think early in the epidemic, I was pretty proud of myself, a little prouder than I probably should have been, considering the teamwork that we really had going on. But, you know, we bring our personalities to our life, you know, and so no huge regrets.
I just want to add one other thing, and that is an absolute key to whatever success that I have had is being able to assemble a great team to support my work. And I would, you know, for example, Robin Givens, who's been my executive assistant for over 20 years. Robin, are you here?
I think he's in the room.
Oh, he's in the room. Oh, there he is.
Let me raise the volume.
Well, you can see that Robin has been my right hand, my left hand, you know, and if you can find that type of relationship in terms of people that you can count on, that you can turn to for advice, et cetera, and that who will enable all of your grand ideas, find them and never let them go. Did you hear that, Robin? Never.
We have some time for a couple more questions.
Welcome, Judy. We're just doing a fireside chat.
You got it.
So, Warner, especially, you've had a chance to work with some really important members of the lay community and people affected by the epidemic, Martin Delaney especially, and others. Could you comment about how those relationships and their ideas help actually even framing some of the questions or hypotheses that you chose to be passionate about?
Yeah. So the question is really kind of the impact of the lay community, in particular the activist community around HIV. How have they helped shape my personal science? And I would say I remember distinctly the dinner that I had with Martin Delaney, the day that we really discussed formation of the Martin Delaney, what would become known as the Martin Delaney Collaboratories. We were in Washington, D.C. to talk about whether we could ever give HIV-infected individuals an HDAC inhibitor. Was it too toxic? And we started, I think we changed the conversation to how can we actually attack this virus in the latent reservoir in a more intelligent way. We need to work together. And that led to the concept of a collaboratory, which is now funded through multiple millions of dollars of NIH dollars. And Martin was he celebrated that. He helped drive it. And in fact, you know, unfortunately he passed away, but it was named after him. I've been struck by how many times you know, that members of the affected community will come and ask a simple question that, oh, wow, I didn't even think about it that way. And that's really changed my perspective. So it's been wonderful having that type of interaction. And it's been, when I worked on the IL-2 receptor or NF-kappa B, didn't have that. It's really this kind of very dynamic interaction with the community has been a wonderful part of the science of HIV.
Let me take it in a little different direction. In the very early days, when we were seeing our first patients, we physicians recognized that we didn't really know anything about the gay community. For some reason, even in San Francisco, there just wasn't any interaction. And we realized that our patients didn't have families, and we had to learn a lot about the community. And what we learned was that there were families and that there were people, organizations, and so what I learned in those days was kind of the rich resource that that represented, and that was obviously we helped support that. I'm not answering your question really about science, but it became really an essential part of how San Francisco responded so well to the challenge that this represented. It required kind of a lot more interaction than there had been. I think physicians are often, you know, you see the patient in front of you, the problem in front of you, and that's it. But I think going back to what we were saying earlier, considering the social setting of now we're talking about HIV, but you know we could easily be talking about the social setting of many other conditions. Is a new direction and one that was really driven by interactions with the community.
I had a question, sort of looking to the future, both of you. Could you give us a sort of an optimistic vision of how things can look for junior investigators? I think there's a temptation to think that it's more competitive than ever to get grants, and we live in this strange geopolitical moment with, you know, certain people in the White House who don't seem to necessarily endorse the worldview of academic institutions like UCSF. So, you know, what's the glass half full for folks like us?
Yeah, no, that's great. So I did a thing for UCSF Homecoming last Friday downtown, which was kind of a fun thing. And I was on a panel, and I was asked a similar question to that, and I was a little bit maybe too negative in terms of, because I think Warner's pointed out the challenge. You know, it is a different world, and I think I may have mentioned that philanthropy for HIV seems to be close to impossible. But Diane Havler was on the panel, and she took it in a really different direction and pointed out all the exciting things that are now in front of us. And you know, I don't really know implementation science. I follow Alvin, and I know it's important, but I think there's going to be just a ton of things that we can do in the spirit of kind of, okay, we got the tools, how do we use them? And I think we're going to have some presentations today that get at that. So I think it's a great opportunity for behavioral scientists and physicians to really kind of sit down together and learn about the methods of implementation science. I think there's going to be money in it too. I think there's going to be funding for that work. And I think the more places like San Francisco can show real progress against the epidemic, we become kind of a beacon for that. So I think this is going to be a spectacular place to be a young scientist. Maybe not at the bench thinking about fundamental kind of control, but.
I think we're at a tipping point in Africa where most infections are occurring. We've made tremendous inroads now, over 20, I think the last number I saw, 21.7 million people on antiretroviral therapy out of the roughly 37 million infected. But there's still significant parts of sub-Saharan Africa which are not, where people are not being able to access antiretroviral therapy. We see that programs like PEPFAR are retracting. We see the Global Fund retracting. I'm worried that there is not the will of the developed world to complete the job of trying to end this epidemic in the developing world. So I worry about that. But that's where the opportunities really lie, as Paul was saying, in implementation science. Let's figure out the way to get this done and get it done. And so, yes, I think for many people, all the tools are in the box. Now, figure out the best way to use those tools to really attack this virus on a global scale. That's the challenge.
Paul, Warner, thank you so much.
Sure.